Hello, everyone. Welcome back to my TCR-seq video tutorials. In my previous video, I showed you how to perform the basic clone-type visualization. Today, in this video, I'm going to show you more functions for the clone-type visualization. If you look at the online tutorial, those functions were defined as more advanced function for clone-type visualization. So the first function I'm going to show you is the colonial proportion. So let's run this function first, and then we can explain the function using the figure. Let's run. You can see we generated a figure in the plot window. So the x-axis shows the 6 samples from this dataset, and the y-axis shows the occupied repertoire space. So the total space will be 1. So the yellow color shows the clone types in each sample that have abundance 1 to 10. The orange color shows the clone types that have abundance from 11 to 100, and the red-orange color shows the clone types that have abundance from 101 to 1000. Then the purple color shows the abundance between 1001 and 10,000. Then in this dataset, we don't have clone types that show abundance more than 10,000. Here, the Colonial index setting is the default setting. You can see this setting was set by the split argument. So the default setting will be 10, 100, 1000, 10,000, 30,000, and 100,000. So if we run this with the default setting, we will generate the same figures here. So let's run. You can see no changes for this figure. So we know the highest abundance in this dataset is 482. So actually you can change the default settings here to have a color range is more suitable for your dataset. For example, this dataset, we can set the upper limit as 500. So with the colonial proportion function, you can also change the clone core to strict, NT, or gene. So let's set the clone core as strict. We can run it again. You can see we generated a new figure. Here the clone core is strict. If you want, you can practice use clone core as NT or gene. So next function I'm going to show you is the colonial homeostasis. Again, we can run this function with the clone core as the gene first to generate a figure. Let's run. You can see we generated a figure. Then the colonial type group, this is the default setting for this function. So for the dark blue color, it was set as rare between 0 to 10 to minus 4. Then for the small group with the purple color, it is between the 10 to minus 4 and 0.001. For the medium group with the red-orange color, it is between 0.001 to 0.01. For the large group is 0.01 to 0.1 with the orange color, and the hyper-expanded group is 0.1 to 1. You can see in this dataset, most clone types are in the small group, and only the tumor sample from the third patient has the clone group in the range of hyperexpanded group. So this orange color shows clone type expansion in the third patients for the tumor samples. So again, this is the default setting for the clonal homeostasis function. We use the Clonotypes argument to set these default settings. If we run this function use the default settings, we will generate the same figure again. Let's run. You can see no changes for the figure. So you can use other clone core variables such as NT and streak. For demonstration, we can use the default setting to run the clone core as amino acid. Let's run. So this figure is for the amino acid clone core. Once again, you can change these settings to make the color scheme is more suitable for your dataset. So now we can move on to see the third function called clone overlap. So the overlap analysis measures the similarity between different samples. In this function, you can use different clone cores for the demonstration. I'm going to use the strict. So there are 4 methods you can use. The first method is the overlap. Let's run this function first. You can see we generated a figure to show the overlap coefficient. You can see we have the clonotype overlap between the tumor and the peripheral blood samples in the same patient. So here is the coefficient for the second patient between tumor samples and the peripheral blood samples. And here is the coefficient for the first patient between peripheral blood and the tumor samples. And here is the coefficient for the third patient between tumor samples and the peripheral blood. Then you can see between patients, the coefficient is 0, which means the overlap similarity for the clonotypes between different patients is very low. So we run the method as overlap. You can also use the method as Jacquard. Let's run. You can see we generated a similar figure as the overlap method, and the Jacquard index number show the overlap between peripheral blood samples and tumor samples in the same patient, but they don't have clonotypes overlap between different patients. The third method that you can use is the Morista method. We can run. You can see here is the Morista index for the tumor samples and the peripheral blood samples in the same patient. Patient 2, patient 1, and patient 3. Once again, there is no clonotype overlap between different patients. So the last method you can set is the raw. If you set the method as raw, here we will show the numbers. Let's run. You can see now we use the raw method. Here we show the total number of clonotypes that overlap between the tumor and the peripheral blood samples in the same patient. So for the first patient, we have 93 clonotypes overlap between tumor and the peripheral blood. For the second patient, the number of the clonotypes is 63, and the third patient is 65. So in addition to use the clonal overlap function, we can also use the clone size distribution method to analyze the similarity between different samples. In this function, for the demonstration, We still use the clone core as strict, but you can practice as gene, amino acid, or nucleotide. So the method we are going to use is ORD2. This is a hierarchical clustering method. We can use this method to cluster the samples by the clone size distribution. So let's run this function. So you can see, using this clone size distribution function, we generated a dendrogram to show the similarity between different samples. You can see here, we clustered the peripheral blood samples together, and here also clustered the tumor samples together, even they are from different patients. So we already know from previous analysis, the peripheral blood samples from the first patients is the outlier sample. So the samples from the first patient was clustered separately from tumor samples and the peripheral blood samples. So next function I'm going to show you is the diversity analysis. This function called colonial diversity. So the diversity is calculated using 5 metrics. The first one is Shannon, and the second one is inverse Simpson. Both metrics estimate the baseline diversity. Then the third one, Chow and ACE. ACE stands for abundance-based coverage estimator. Chow and ACE metrics estimate the richness of the samples. And the last one is the inverse P&O. These metrics measure the species evenness. Here we actually measure the clonotype evenness. So let's run the clone diversity function. We can use the clone core as gene, and the group by function as samples, and the x-axis as ID to demonstrate this function. So let's run. You can see we show the analysis for 5 metrics: Shannon, inverse Simpson, Chow, ACE, and inverse P&O. So the Shannon and the inverse Simpson estimate the baseline of diversity. You can see basically the tumor samples has lower diversity than the peripheral blood samples. Chow and ACE estimate the richness of the samples. Once again, you can see in the peripheral blood samples show much higher diversity than the tumor samples. So the last matrix, inverse the P&O, measure the species evenness, and the tumor samples has a higher value, again, which means the tumor samples has a lower clone type diversity. So from the colonial diversity analysis, we can see the tumor samples has a lower clone type diversity than the peripheral blood samples. So the last function I'm going to show you for the advanced visualization is the scatter clone type function. Using this function, we can directly compare clone types between 2 samples. For example, here we can use the clone core as a gene. Then we set the x-axis for the peripheral blood samples from the first patient, and the y-axis as the tumor samples from the first patient. So basically the x-axis and the y-axis are used to set the 2 samples you want to compare. Then let the dot size argument, we can set it as the total. If we set it as the total, the figure will display the total number of the clones In both samples, we set in the x and the y axis. Then the graph, we can set it as proportion. You can also set the graph as count to show the numbers from both samples. So let's run this function as the last demonstration. We can run. So you can see we generated a scatter plot. The circle size indicates the number of total clones, and the yellow color shows dual expanded clone types. So which means those clone types are detected in both peripheral blood and tumor samples. If you see the purple color, those clone types are tumor specific, and the orange color are the clone types that are only presented in the peripheral blood samples. Okay, this is the demonstration for advanced visualization of clone types. In my next video tutorial, I start to show you how can we interact the TCR-seq data with single-cell mRNA sequencing data. So I hope to see you in my next video tutorial. Please don't forget to subscribe my channel and share my videos with your friends. Thank you and see you next time.